When we look at diagnosing bacteria, there are a couple of things that become very interesting. Number one is, since bacteria are very tiny and they're single cells, they often accumulate inside of plant tissues. When they do that, if you break the plant tissue and if you pull it apart, many times you will see a stringy or a mucousy type format in here. That might give you an idea that you've got a bacterium. What they did on the top was they took a little piece of leaf tissue off of this leaf, they put it in water, then they looked at it under the microscope. When you do that, you see the bacteria streaming out of it. This is something that you can actually do without even needing a microscope. You just take that piece of leaf tissue, you put it in a glass of water halfway submerged, hold it to the end, look through it, and you would be able to see the masses of bacteria coming out. These are somewhat diagnostic that you have a bacterial problem. When we deal with bacteria, one of the things we always deal with is some of them will have vectors. This is a type of what we kind of call a phytoplasma. It is transmitted by this glassy wing sharpshooter that's on your right-hand side. These are quite problematic in things like grape. The only way you get them transmitted is through the insect. If you wanted to control this particular disease, one of the things you try doing is you try killing off the insects to keep them from transmitting it. This is a wilt on a form of cucurbit. What you see here is the plant has been infected by a bacterium. As it did that, the plant itself has crumbled and is no longer viable. This is the inside of a potato with a rot in it. And this, believe me, this looks bad. You should smell it. It smells much worse than it looks. There are certain bacteria that are problematic. They will liquefy the entire organ that they get into. A lot of times when you find bacteria in your refrigerator, they really have a foul odor associated with them. Cabbage. You can see the edges of the leaves where the hydothodes are. This is where the bacteria got in. This particular one is Xanthomonas campestris. It's very common on things that are in the cruciferous family, such as cabbage and broccoli and cauliflower. This is a close-up of a broccoli leaf. You can see where this symptomology started at the very edge of the leaf. This bacterium was getting into the hydothodes. That's a very big problem with bacteria when we find them, is they often do start at places like the hydothodes. This is tomato. This whole tomato crop's been wiped out by bacteria. I'm not sure you can really do anything with the tomatoes that are there. They're all going to have some sort of material on the inside that's going to render them virtually useless. This is a tomato with a scab on the outside. This is caused by a Corynebacterium. The one thing about this symptomology, it's superficial. It many times does not progress inside of the tomato itself. These are crown galls. The one on the left-hand side is on a rose bush. You can see the size of the gall compared to the size of the plant. It's quite large. The other one is also on a woody plant. This bacterium that causes this is very interesting. They've actually used it for transmitting genes into plants. Whenever we look at bacteria, one of the things we have to talk about is nitrogen fixation. There are several types of organisms that fix nitrogen, convert atmospheric nitrogen to nitrogen in a plant available form. They all are bacteria. So we've got nitrogen fixing nodules. This is a symbiosis between a bacterium and a plant root that allows for the bacterium to fix the nitrogen. The bacterium gives the nitrogen to the plant gives the bacterium sugars in return. Many different types of bacteria. They all are in the major genus of Rhizobium. Rhizobium has since been broken down into a lot of different types. This shows you a rhizobium on a root. You can see that these are three-dimensional structures. They are pink in color. The pink in color indicates that they are actively fixing nitrogen. These will take nitrogen out of the air and convert it into a biologically available form that will be picked up by the plant roots. Here's nitrogen nodules on soybean. This is a nitrogen nodule on another type of plant. You can see how they get to be rather prevalent all over the root system of the plant. These are another set of nitrogen nodules. Some bacteria that we have actually reside inside of the phloem. This particular one is inside of the phloem. It is what we used to call a mycoplasma, which means it really doesn't have a shape like a coccus or a bacillus, but rather it's got a free-form shape that allows it to move around. These get to be quite problematic in plants because they will block up the phloem. If they block up the phloem, that is going to cause severe handicap to the plant. one of the things we always look at is how do these things get into tissues? One methodology is direct. We talked about that with fungi, where the fungus goes and penetrates directly. We have one group where they get in through the stomata. The stomata are little openings on the outside of the plant. You can see a stomata right there and the substomatal cavity underneath of it. When that's open, anything can migrate in. Over here, we've got a top view of stomata. You can see the stomata there. They can get in through the hydrathodes. We talked about the hydrathodes being the outer end of the vascular tissue. What happens is as the plant is nearing the end of the day, it's been pulling up moisture all day long. It stops needing all that pull-up of moisture. It takes it a while to shut down the pressure of pulling the moisture up. While it's doing that, moisture will actually go out the ends of the vascular tissue in what we call the hydathode. They will remain on the outside until something knocks it off. If nothing knocks it off, in the next day, these plants will often pull the material back in. If they pull that material back in and there's a bacterium sitting in that material, that bacterium is going to get in through the hydathode. If it gets in through the hydathode, then it's going to be able to transmit and start a problem. Here is a leaf. You can see how this symptomology started at the junction of the vascular system with the outer margin of the leaf. We have wounds for plant material to get in. This is sap sucker damage on a tree. What the sap sucker does is it comes around and it picks these little holes in. This causes sap to come out, or nutrition in the phloem to come out. That will attract insects. Then the sap sucker will come back. It'll look for the insects and pick them out and eat them. These serve as an area where something can get into the plant. If they get into the plant like that, they can be quite problematic. This is another very interesting thing with certain kinds of bacteria. When you observe bacteria, many times they will exude out of the plant tissue onto the outer part of the leaf where they might dry down. Then it kind of gives you a glazing on the outside of it. It kind of resembles the glazing on a donut. You've got this polysaccharide type material outside of the bacterium. It kind of glosses over the outside of the plant. When we look at bacteria, a lot of different things we can look at, a lot of different things we can talk about. But bacteria are very interesting.